Subscribe to Simplified Biology channel and press the bell icon to get notified when a new video is uploaded. Hello friends, welcome to Simplified Biology. Today's topic is auxins, the plant growth hormone. Auxins. The term auxin means to grow. Now, auxin is a weak organic acid first discovered from human urine in persons suffering from pellagra, that is deficiency of niacin, nicotinic acid. It is found present at the shoot tip and the root tip. It shows polar movements, that is from shoot tip downwards and root tip upwards. The precursor of auxin, means from which it is synthesized, is the amino acid tryptophan. And the metal that is used in the synthesis of auxin is zinc. The site of synthesis of auxin in the plants are young leaves, shoot apices or the shoot tip, root tips, and developing seeds. Charles Darwin and his son Francis Darwin first reported the presence of auxin in the coleoptile, that is the shoot tip of canary grass, that is phalaris. He showed phototropism, that is growth towards the light. Now, when the shoot tip or the coleoptile of canary grass was illuminated or light was given from one direction, the shoot tip started growing towards the light. That is, it showed phototropism. Now, when the tip of the coleoptile was removed and it was illuminated, no growth occurred, or it did not show phototropism. Next, Wendt performed the agar block experiment in Avena coleoptile, that is in the shoot tip of oat. Wendt took an Avena coleoptile, decapitated it, that means the tip of the coleoptile was removed. This tip was kept on an agar block for a certain period of time. After which, instead of replacing the tip on the coleoptile, he replaced it with the agar block. He kept the agar block towards the darker side of the coleoptile or the shoot tip. He saw that when this tip with an agar block kept on its darker side is illuminated or light is given from one direction, it started growing towards the light. Now this showed that something is being synthesized at the tip, that is translocated downwards towards the darker side and is responsible for growth. Wendt isolated it and named it as auxin. Natural auxins are indole acetic acid and indole butyric acid, that is IAA and IBA. Synthetic auxins are 2,4-dichlorophenoxyacetic acid, that is 2,4-D. Naphthalene acetic acid, NAA, while indole butyric acid is also used as a synthetic auxin. Auxins are widely used in agriculture and in horticulture, that is during cultivation of flower, fruits, vegetables and ornamental plants. Physiological effects and uses of auxin. First, it causes growth by cell enlargement, that is by increasing elasticity of the cell membrane. cell wall, increasing permeability of the cell membrane, and increasing rate of respiration. So it is capable of causing cell enlargement by increasing elasticity of the cell wall, increasing permeability of cell membrane, and increasing the rate of respiration. Second, it has no effect on root growth. It can cause growth of the shoot but has no effect on the root growth, but can produce new roots. So it is capable of producing new roots but has no effect on root growth, due to which it is used in tissue culture for the formation of plantlets. Now, this property of auxin is also used in vegetative propagation of stem cuttings by stimulating root formation. So, in this diagram, you can see this is a stem cutting in which the root formation is promoted by the help of auxin. Third, it increases cambial activity by inducing cell division in the cambium. This is used in tissue culture for callus formation. So, auxin is capable of causing growth by cell division and cell enlargement. Next is apical dominance. That is, the apical bud prevents the growth of lateral buds. Now, as the apical bud has a high concentration of auxin, it prevents the growth of the lateral buds. These lateral buds, they are unable to grow or they become dormant due to the presence of the apical bud. Now, when the apical bud is decapitated, or you can say pruning is done, means the apical bud is removed, the lateral buds, they start growing. Lateral buds start growing, giving the plant a bushy appearance. This property of apical dominance is used in hedge making, in gardens, and in tea plantation. In tea plantation, where the leaf is used for making tea, the more the leaves in the plant, the more is its value. So, since apical dominance to prevent the growth of the lateral buds, pruning is done so that the trees or the plants have a bushy appearance and have more leaves in it. Apical dominance is also used to prevent sprouting of potato tubers in storage. Potato tubers have buds on it and they start sprouting, decreasing the food value of potato. This is prevented by applying auxin to them. So they make the potato tuber dormant, hence preventing sprouting of the potato tubers. Next, it prevents abscission of young leaves and fruits by preventing the formation of abscission layer. Abscission means falling off of young leaves and fruits. By preventing the formation of abscission layer. Abscission layer is a separation layer formed at the base of the petiole, after which falling of leaves and fruits occur. Now, this is prevented by auxin, and this is used to prevent falling off of fruits in apple, orange and tomato. Next, auxin normally inhibits flowering, but it promotes flowering in pineapple and lychee, due to which uniform flowering occurs, leading to fruit formation, and it delays flowering in lettuce. Next, parthenocarpy. Parthenocarpy is formation of fruits without fertilization. Auxin is capable of inducing parthenocarpy by application of auxin to an unpollinated stigma. Application of auxin to unpollinated stigma induces parthenocarpy, that is formation of fruits in tomato, banana, cucumber. These fruits are seedless. Next, eradication of weeds. Weeds are unwanted plants. And they are usually the dicots or the broadleaf plants. Auxin is sensitive to dicots but has no effect on monocots. Now, when for example, in a crop of cereals are the monocots, where the weeds are growing, when 2,4-D is sprayed, it causes excessive, uncontrolled growth in the weeds or the dicots, killing them without affecting the cereals. So they are capable of removing weeds without affecting the cereals. That is why they are used as weedicide or herbicide. They are used to prepare weed-free lawns. And last, auxin also controls xylem differentiation, that is formation of xylem elements. So, auxin is capable of causing growth of the shoot by cell division and cell enlargement. It has no effect on root growth but forms new roots. It shows apical dominance, that is, the apical bud prevents the growth of lateral bud, and when the apical bud is removed, a bushy plant can be formed. Next, it prevents abscission, that is, falling off of leaves and fruits. It promotes flowering in pineapple and lychee. It is capable of inducing parthenocarpy, that is formation of fruits without fertilization. It is capable of eradicating unwanted plants, that is weeds, and last, it controls xylem differentiation. So these are the physiological effects and uses of auxin. So that's all for today. Please do like, subscribe and comment, and also visit our website by clicking on the link given below. Thank you for watching.